The purpose of Prentice Hall's video tutor is to guide you through difficult anatomy and physiology concepts with detailed 3D animations and video footage. While you're reading through fundamentals of anatomy and physiology, you'll see these icons in certain sections. This means that a supplemental explanation is provided in the video tutor. Once you start watching the tape, view each section without stopping. Then go back and watch each section again. This time, stop the tape and review it as many times as needed. And if necessary, watch the entire section for a third time. Then answer the study guide questions using the video tutor as a quick reference. Prentice Hall, in conjunction with Mimi Brass of Education Resource and Blosson Medical Communications, has produced this video tutor to supplement your anatomy and physiology textbook and lecture notes. The tape was developed to complement fundamentals of anatomy and physiology by Frederick Martini, but the video tutor doesn't have to end here. You'll find it very useful no matter what text you're studying. The concepts you'll learn about in the next 30 minutes will also apply to related courses like anatomy, physiology, and introductory biology. Sometimes visualizing physiological processes can be difficult. The text provides a solid foundation, but couple that with detailed 3D animation and you're off to a good start. Video footage is also implemented to further explain each concept. And your video tutor host will provide easy-to-understand explanations and examples. Each concept you learn will start with a broad overview and then works its way from a macro down to a micro view of the body system. And when you've completed each section, answer the questions in your study guide to assess what you've learned. Hi, I'm Suzanne Savoy, and I'll be your host for the video tutor discussion of membrane transport. Cells come in different shapes and sizes. They may be tall and thin or wide and flat. No matter what the shape and size, most cells have similar structures. The cells of your body are like rooms in a house. Just as each room in your house is surrounded by walls, every cell is surrounded by a thin membrane. It is called the cell membrane. The cell membrane is extremely thin, but it's still a complex structure of lipids and proteins. The arrangement of proteins and lipids is fluid or changeable, and the proteins drift from place to place within the membrane. The cell membrane is mainly composed of phospholipids. Each phospholipid molecule has a head and a tail. The head is called hydrophilic because it has a polar structure and is attracted to water molecules. The fatty acid tail is called hydrophobic because it is nonpolar and it repels water molecules. Cell membranes have two layers of phospholipid molecules. The hydrophilic heads face outward and are exposed to the water. The hydrophobic tails face inward and are shielded from the water. This structure is called a phospholipid bilayer. The lipid bilayer gives the cell membrane support and creates a barrier between the cell and its environment. Most solutes are unable to cross the membrane, but the membrane is not completely sealed, just as the rooms in your house are not completely airtight. Similar to the way air seeps through cracks in your walls and windows, some substances can slip through the cell membrane. Some molecules move across the membrane by diffusion. Atoms and molecules are constantly moving. Just like bumper cars, they collide and ricochet off each other. When bumper cars collide, they scatter into a less crowded area. They spread out from an area where they are highly concentrated to an area where it's less crowded. This is similar to what happens to particles inside our bodies. First, let's define some terms that deal with diffusion. When these particles are dissolved in fluids, they're called solutes. The difference in solute concentration between two areas is called a concentration gradient. And lastly, the net movement of solutes from an area of high concentration to an area of lower concentration is called diffusion. You can better understand the concept of diffusion by watching this ink drop. The ink molecules are highly concentrated in the drop, but not concentrated anywhere else in the glass. The ink molecules in this glass behave like bumper cars. The ink and water molecules collide with each other, and the ink molecules gradually diffuse outward from the drop. They follow the concentration gradient. So the ink molecules are moving from an area of high concentration to an area of lower concentration. The ink molecules continue to scatter until they are spread evenly throughout the glass. Even though membranes weren't involved in the ink drop example, diffusion can also transport molecules across a cell membrane. Diffusion is a transportation bargain for your cells. They don't have to expend any ATP energy to make molecules enter or leave by diffusion. Molecules can diffuse across a cell membrane by two methods. Fat-soluble molecules such as steroids, alcohol, and some vitamins can dissolve in the lipid portion of the membrane. Water-soluble molecules cannot cross the membrane without channels. Cell membranes have two-way doors that allow molecules to enter and leave the cell. These doorways are formed by various types of proteins that float in the fluid membrane. To further explore this comparison, let's look at different types of doors. Some doorways have no doors, so they're always open. Others have doors that open and close. Still others let people move in and out at the same time. As we learn about cell transport methods, we'll see examples of all these types of doors. Channels that are always open are called leak channels. Those with doors that open and close are called gated channels. Leak channels and gated channels are very specific. Only solutes of a particular type can pass through a given channel. When a gate is open, molecules can diffuse through the channel freely, following the concentration gradient. No ATP energy is needed to move them. When water diffuses across membranes, the process is called osmosis. Osmosis of water molecules occurs in the same way as diffusion of solute particles. The water molecules move from an area of high water concentration to an area of lower water concentration. Osmosis occurs across a membrane that is freely permeable to water, but not to solute particles. This glass contains two solutions with different solute concentrations, separated by a membrane permeable only to water. This membrane in this demonstration behaves in a similar way to your own cell membranes. Solution B on the right has a higher concentration of solute molecules than solution A on the left. Solution A on the left has a higher concentration of water molecules than in solution B, since less space is occupied by solute molecules. The solute molecules are too large to pass through the pores in the membrane and must remain on their side of the barrier. But water molecules can move across the membrane. The highest concentration of water molecules is in solution A, since there are fewer solute molecules on that side of the membrane. Water molecules move across the membrane from the area of high water concentration, solution A, to the area of lower water concentration, solution B.
As the water moves across the membrane, the volume of solution B increases. Water will continue to cross the membrane until the solute concentration has been equalized or until further movement is prevented by an opposing force such as gravity.
Note that osmosis forces water across the membrane into the solution with the higher solute concentration. The difference in solute concentrations between the solutions is called an osmotic gradient. The tissue fluid that surrounds your cells is called isotonic because it will not cause a net movement of water into or out of the cells.
An extracellular solution is called hypotonic if osmosis will force water across the cell membrane and into the cytoplasm. The cell will swell, as in the case of this red blood cell. Eventually it may burst, an event known as hemolysis.
An extracellular solution is called hypertonic if water will flow across the membrane and out of the cytoplasm by osmosis. The cell will shrivel and dehydrate, a process called crenation. Most molecules needed by cells are not lipid soluble and they're too large to diffuse through membrane channels. These molecules make use of a system of carrier proteins embedded in the cell membrane. Carrier proteins function like revolving doors. They pick up molecules on one side of the membrane and release them on the other side. Each carrier protein can transport only a specific type of molecule across the membrane. Some molecules that are too big to pass through channels may be passively transported across the membrane following their concentration gradient. This process is called facilitated diffusion. Facilitated diffusion transports various sugars, amino acids, and nucleotides into and out of cells. The molecules are still traveling with the concentration gradient, and ATP is not required. Some transport mechanisms require ATP. These are called active transport mechanisms. During active transport, carrier proteins move solute molecules across the membrane. The direction of movement is independent of any existing concentration gradients. Using active transport, cells can accumulate solute molecules at much higher concentrations than are present outside the cell. The sodium-potassium exchange pump is an important example of active transport. It is active in all your cells.
This exchange pump transports sodium ions out of your cells in exchange for potassium ions. Both ions are moved against their existing concentration gradients.
The sodium-potassium exchange pump is important in maintaining differences in ion concentrations between the intracellular and extracellular fluids. After viewing this material on membrane transport, you've seen that all cells are enclosed by phospholipid membranes. Some molecules move freely across the membrane, others pass through channels, and still others are assisted by carrier proteins. So the next time you pass a doorway, remember the different types of doors found in cell membranes and try to imagine which type of cellular door you are passing through.
Hi, I'm Suzanne Savoy, and welcome to the video tutor discussion of protein synthesis. By now you know that the nucleus is an information storage center, and DNA molecules hold this information. In order to understand protein synthesis, you can think of the nucleus as the headquarters of a manufacturing complex. To produce a product, you need instructions, and DNA contains all of the necessary information. DNA molecules are made of chains of nucleotides. Two long strands of nucleotides wind around one another like a twisted ladder. This structure is called a double helix. The sugar phosphate backbones of the nucleotides make the sides of the ladder. The rungs are made of pairs of 4 different nitrogenous bases: adenine, guanine, cytosine, and thymine. The pairing of these bases is very specific. Adenine always bonds with thymine, and cytosine always bonds with guanine. This is known as the base pairing rule. Each DNA strand contains thousands of base pairings. A strand of DNA, along with its regulatory proteins, is called a chromosome. We all have 46 chromosomes. They contain instructions to make all the necessary proteins in the body. The nuclei in our cells have information to synthesize over 100,000 different types of proteins, and each cell has a copy of this information. A gene is a portion of DNA that has all the needed information to make one polypeptide chain. A single protein may consist of several polypeptide chains. DNA molecules don't leave the nucleus or build proteins directly. Instead, the process is carried out by 3 different types of RNA molecules, which work together to control protein synthesis. The RNA is a single-stranded nucleotide chain containing the same nitrogenous bases as a DNA molecule, except for one. It has uracil instead of thymine. To understand how RNA is involved in protein synthesis, we go back to the analogy of a manufacturing company. To produce your product, you need a blueprint of the instructions, raw materials, and a factory to assemble the components. Each type of RNA does one of these 3 separate jobs. Messenger RNA contains the blueprint to build the product. It carries the information from the headquarters or the nucleus to the factory. Transfer RNA pick up and deliver the raw materials needed in the factory. Ribosomes, made of ribosomal RNA, are the factories that build the product. Now that we know the responsibilities of each molecule, let's take a closer look at each step in the process. Protein synthesis is divided into 2 stages, transcription and translation. During transcription, the genetic code is transcribed or copied in the nucleus. It goes from the DNA molecule into a messenger RNA blueprint. During translation, the messenger RNA molecule leaves the nucleus and enters the cytoplasm, where the polypeptide chain is assembled. Now let's explore the steps of transcription and translation in more detail. During transcription, a segment of DNA uncoils and exposes the instructions to make a single polypeptide chain.
A messenger RNA molecule is then created by the enzyme RNA polymerase, which splices together the appropriate RNA nucleotides.
Remember the DNA base pairing rule while we examine the sequence of nucleotides constructed by RNA polymerase. As the messenger RNA molecule forms, a C moves into position whenever the DNA template has a G, and vice versa.